All right, this is our review for Lab 12 in the AP Biology Lab Manual, and that is this is one of the first labs that we actually did, however. This has to do with ecology, so this is Lab 12, the last one we'll talk about. It is called Primary Productivity in the Lab Manual. I'm going to take longer with this one because I want you to really understand the lab. They do ask about it. And also I want to at least go over what ecology concepts I can because we haven't talked about them in a while. So, of course, all food on Earth comes from autotrophs, okay? Whether they are chemosynthesizers in the bottom of the ocean at a deep sea vent, converting inorganic compounds into organic compounds, or whether they're the ones we're familiar with, the photosynthesizers up here, converting light and inorganic CO2 and water into organic sugar that we can then eat. All food on Earth comes from them harnessing something that we heterotrophs can't harness. And putting it into an organic form for us. And of course, because these guys are producing our food for us, we call them the producers, okay? All of these producers, or perhaps I should say at least the ones that are photosynthetic, which is the ones we'll be talking about in this lab, require sunlight, CO2, and water to make food, which is glucose. So this is what they're producing. Incidentally, they're also producing oxygen, but that's not really what we're interested in. Well, I shouldn't go that far either, but normally we're focused in on that glucose. All right. Now, if we want to, as ecologists, to determine the quality of an ecosystem, one of the things we might be interested in measuring or determining is how productive that ecosystem is. That is, how much organic compound, sugar, is getting made in this ecosystem by the producers. That is one thing we can measure that will tell us about the overall quality of the ecosystem that we're studying. So primary productivity can be really important. So primary productivity is one way of measuring an ecosystem. What it is measuring is how much, at least at its heart, is how much food the producers are producing. Okay? And what we mean, in perhaps a slightly more technical term, is how much of that energy of sunlight that those plants are receiving, or algae, is actually getting converted to organic compound. Okay? The reason we care is because how much these producers are producing determines how much life can be supported above them in our energy pyramid, so to speak. Okay? So how much production the producers do determines how much life can be supported above them, because we are depending on them to get our organic compounds. Okay? Now, to determine primary productivity, you have to be able to measure photosynthetic rates and cell respiration in the ecosystem that we are studying. Okay, that's at the bottom of it as far as what we need to be able to measure: photosynthesis and cell respiration. Okay, now if we want to measure photosynthesis and cell respiration, remember their equations. Okay, photosynthesis, if you'll recall, is light plus CO2 plus H2O is going to give us glucose plus oxygen. Okay? Now, we could simply measure the sheer production of glucose to determine how much photosynthesis is occurring. We could also measure the production of oxygen, because that would also tell us how much photosynthesis is occurring, because this is also a product of that process. We could, if we wanted to measure respiration, on the other hand, remember respiration is glucose plus O2 plus, gives us CO2 plus water plus energy in the form of ATP. So, if we wanted to measure cell respiration, we could measure the production of CO2. We could measure the production of water, though that would be tricky. We could measure this, but that would also be tricky. CO2 is our easiest thing to measure if we're measuring production. We could also measure how much of certain things gets used up if we wanted to determine rates of photosynthesis and cell respiration. Now, let's think of an aquatic ecosystem. Why? Well, it's an easy one to measure, okay, in terms of photosynthesis and cell respiration. So that's why we do that in this lab. In such an ecosystem, one of the easiest things we can measure is production of oxygen and use of oxygen, okay? That's very easy for us to measure. We have a little dissolved oxygen meter. We can measure the oxygen in the water. So let's think about this. The more oxygen in water over a period of time, we can conclude that there's more photosynthesis being carried out by the producers. If the oxygen's going up, then somebody's putting it there. It must be the producers, okay? That directly translates for us in, well, if there's more oxygen, there must also be more food available. And so this must be a reasonably productive, independent ecosystem. Now, in a lab, if we're measuring productivity, that's great, but what we always need an independent variable. What are we going to manipulate about this? Well, in this lab, what we were asked to manipulate was light intensity. Light intensity simply means how much light are we shining on that thing. Is it dark? Is it shaded? Is it full light? Are we just, whatever. That's light intensity. So, what would affect the ability of light to be available in an aquatic ecosystem? Well, surely you would understand that if this is a pond, there might be more light available at the top and less light available at the bottom. So that's a really real variable that would be at play in an aquatic ecosystem. Okay? Can light be a limiting factor? Remember the term limiting factor, something that's going to limit productivity, limit population growth. Light can be a limiting factor on land too. In a shady tropical rainforest, we've got a tree, and then this little plant that's trying to grow down here can't get light because he's being shaded. So certainly it can be a limiting factor there, but certainly in the water as well. Okay? Now, let's say you have an aquatic ecosystem in front of you. Okay? Let's say it's my aquarium in the back of the room. Okay, and if you've looked at my aquarium, you know it's a big green mess, but I think it's a big green beautiful thing because it's got all kinds of producers in there going crazy. The producers in that ecosystem are algae. Okay, there's actually a few plants in there, but mostly it's algae. Some of it's called Spirogyra. All of this is Protus chlorophyta green algae. Okay. Now, what would affect the ability of my algae to produce? Well, the amount of light available to them to do so, the amount of CO2 available to them. We know if we shut off their CO2 supply, they're going to be in trouble. There are all kinds of things that could affect their ability to produce glucose. Now, are there consumers in there? Well, I can tell you that there are. There are some tiny little crustaceans in there called Daphnia that are swimming around. There's other stuff that's like unicellular, like Paramecium. They're in there. Okay, so there are consumers in there as well. Now, our experimental question then is, what is the effect of light intensity on the primary productivity of a freshwater ecosystem? Okay, is light going to affect the amount of photosynthesis? Now, hypotheses we could suggest for this? If light increases, then primary productivity will increase. If light decreases, then primary productivity will decrease, whatever you want to think of. Now, let's talk about experimental design. Our independent variable, then, is the amount or intensity of light. Our dependent variable that we will measure is we are actually going to measure how much oxygen is in that water. The more oxygen, the more productivity. So our dependent variable really that is meaningful to us is productivity. But how are we getting at that? We're measuring literally the milligrams of oxygen in the water. Okay? So now, experimental treatments. What this means is we will have a control, and then we will have other treatments that are getting some version of the variable. Okay, what will they be? Will we have a bottle that is in the dark? Will we have a bottle that is in full light? Will we have a shaded bottle? What will our experimental treatments be? And which of these will we consider the control treatment? What are things that we need to hold constant between all of our different treatments? Maybe the amount of water in each treatment, maybe the amount of, maybe the temperature of the environment. Maybe we would want to hold that constant. The source of the water that we're getting, we would want to maybe hold that constant, and so on. And of course, if we could, we would want to do several trials to make sure that if there is an error, that is not the foundation for our result that we are going to conclude from. Okay, so here's what you would do. In this case, we're going to obtain water samples from our aquarium in the back. 3 different bottles of water samples. And what we do then is measure the initial level of dissolved oxygen in that water. So we're going to have an initial measurement. We'll call it I. Okay? And then we would seal up those bottles so that the oxygen amount cannot just change because some of it left or new oxygen came into the bottle. We want to lock in what's there and then be able to measure it at a later time. So we seal up those bottles, then expose them to different amounts of light for 24 hours. Okay? Our experimental treatments will be dark, no light, shade, half light, and full light, 100%. And then we'll simply measure the dissolved oxygen in those containers after 24 hours. That's our basic procedure for this primary productivity lab. Now, there are 2 ways to analyze the data that we collect. We can determine the gross primary productivity of our bottles, and we can determine the net primary productivity of our bottles, or our ecosystem, if you will. Okay? Gross primary productivity. You should at least understand what this is. It is the entire photosynthetic production of organic compounds in our ecosystem. This includes all of the glucose made by all of the autotrophs in that bottle, all of them. Okay? And this includes what is stored in their actual tissues and whatever food they might have used up during their own life, doing their own life processes. In other words, when a plant's doing photosynthesis, it doesn't just make the sugar and retain it all to pass up the food chain when it gets eaten. It certainly does store some of that sugar in its tissues, but it also uses up some of that sugar to do its own life processes. So, gross primary productivity is taking into account not only what they store in their tissues, but also what they use during that period of time. And primarily they use that up in cell respiration, doing things to fuel their own life processes like growing, reproducing, etc. Okay? All right. Now, please remember what respiration is. Food material gets made but is used by the organisms themselves during, glycolysis, Krebs cycle, electron transport to make ATP. Now, net primary productivity is simply talking about the organic compounds that remain fixed into organic compounds after we take away whatever the autotrophs have used. In other words, we're not going to count. When you go to work, and please don't use this in an essay question, they do not care about your paycheck. But if you have a job, you work for a week and you get $200, okay? Well, you don't get $200. You maybe get 150 of that. Why? Because you have to give some of it back in taxes. So this is your gross pay. This is what your boss thinks he paid you, but this is your net pay, what you actually got. Net primary productivity is what is actually available to those consumers above the producers in the food chain, not taking into account whatever the producers actually used for their own purposes, because we really don't care about that, because honestly that's not going to get passed up the food chain. That's been used by those autotrophs. So net primary productivity is the gross productivity that we talked about in the previous slide minus whatever got used in cell respiration by those autotrophs. Okay? Now, so which of these tells us what we really want to know? What we want to know is how much organic material is available to the rest of the ecosystem in order for that ecosystem to be productive. And so that is given to us by net primary productivity. Okay? How much organic material is left to go up the food chain is net primary productivity. So we are going to measure the amount of dissolved oxygen present initially. And so then after we do that, we're going to put one bottle in the dark, one bottle in half light, one bottle in full light. And then as we said, we're going to measure the amount of dissolved oxygen in each bottle after 24 hours. If you'll recall, we had a dissolved oxygen probe to do this attached to the LabQuest. And so once we do that, once we take our measurements after 24 hours, then we do a few little calculations and we can determine the cell respiration rate in that bottle. We can determine the gross primary productivity and the net primary productivity. Okay? Dissolved oxygen, we've talked about that. All right. There are many things that can affect dissolved oxygen. Biotic factors, these are terms you might want to remember. Biotic factors are living things, the plants in the water or algae in the water, the animals in the water, cell respiration. The temperature of the water can affect the dissolved oxygen, and that abiotic factors, the amount of light, because it can affect that. Temperature. Hot water, warm water holds less oxygen than cold water does. Cold water can typically hold more. So there are all kinds of things that can determine how much dissolved oxygen can be in water. Okay? Now, when it's time to do our calculations. Let's say we've collected our water samples, we've held them for 24 hours, and now we're measuring and we've got our results. We know how much dissolved oxygen was present after 24 hours. We do some calculations. We do the initial bottle dissolved oxygen minus the dissolved oxygen in the dark bottle. In the dark bottle, the only thing that could happen is respiration. No light was shining on it, so no photosynthesis could happen. The only thing that could happen in the dark bottle is that the organisms in there, little animals, even the plants, the only thing they can do is respiration because there's no light. So we take our initial reading minus the dark bottle reading, and that's going to tell us how much how much cell respiration was going on. Okay? Now, if we take the dissolved oxygen in a light bottle, that's any bottle not kept in the dark, whether it's the 50% bottle or the 100% bottle, we're just going to call those the light bottles. The dissolved oxygen in the light bottle minus that in the dark bottle gives us the gross primary productivity. So light minus dark gives us gross primary productivity. Okay. Now, our third calculation, if we take the light bottle and simply subtract our initial dissolved oxygen that we measured before we left them out for 24 hours, that's going to give us our net primary productivity. Okay? This is going to tell us how much can actually of our production actually is available to get sent up the food chain. Okay? So we've already talked about the dark bottle, how there's no photosynthesis, but there is cell respiration. So we would expect the dissolved oxygen level in there to decrease after 24 hours because the only thing that can happen in there is the stuff, is that cell respiration occurs. There's no photosynthesis, okay? Oxygen would be used up. So if we had an initial reading of 4, and then we had a final reading after 24 hours in the dark bottle of 1 milligram of oxygen, then we would know that we had consumed 3 milligrams of oxygen in 24 hours, and that would be the respiration rate. Okay? Now, we hope in the light bottles at least there's some photosynthesis happening, some productivity. Okay? Okay, but of course there's also cell respiration occurring in there as well. So at any rate, we've got our net primary productivity is going to equal the gross minus the respiration. So which bottle of the 3 should have the least oxygen after 24 hours? Well, of course, the 0 light should have the least. Which bottle of the 3 should have the most oxygen? Of course, the one that had 100% light. Let's see if that works. Okay? We've already been through this. You already been through that. If I were you, I would try to remember those. They sometimes ask about them. So let's say that these are our results. Initially, okay, oh, there it is. Initially, we have 4 milligrams of oxygen. So after 24 hours, in our 100% light we had 10, in our 50% light we had 7, in our 0% we had 1. So that means if we want to do our gross primary productivity, light minus dark is going to equal gross primary productivity. So in the 0% bottle, 1 minus 1, which is the dark bottle, is, so essentially we're doing a dark bottle minus dark bottle because we're calculating for the 0%, is 0. So it was not productive. And our 50%, we're going to take our 7 for the 50% bottle minus what was in the dark bottle, and so our gross primary productivity was 6. And our 100%, we're going to take our 10 minus 1, and that gives us a measurement of 9 milligrams. So this is our gross primary productivity. Okay, 9 milligrams of oxygen, 6 milligrams, and of course 0, not productive. Now, would we expect our net primary productivity to be greater or less than gross? Okay, I would think less because, again, we aren't allowing ourselves to count whatever plants used by themselves to do photosynthesis. So, for each of your bottles exposed to light, we're going to do light bottle minus dark bottle is the gross primary productivity. Okay, we've already done that. Now, we are going to do our net primary productivity, which is the light bottle minus the initial bottle. Okay, so again, if in our 0% bottle, 1 minus the initial was 4, so we have -3. So we can see that really we are losing ground here. Lots of productivity was used up in respiration. 50% light. We've got our 7 milligrams minus the 4 that we started with, and so our net is 3, so it's moderately productive. And in our 100%, 10 minus the 4 we started with, gives us 6. So as you can see, net primary productivity is less than gross, and we can see that productivity in the 0% light is much less than in the 100% light, which is exactly what you would expect. Okay? We would graph this data. We would use a line graph because we've got numerical quantities on each axis that we can plot. Okay, our units, primary productivity was in terms of milligrams of oxygen. Our amount of light was in percent from 0 to 100. Our scale would need to be something like that. You remember you need to put in an appropriate scale. Okay, the title, the effect of the independent variable on the dependent variable. So the effect of the amount of light on productivity is always a nice title. And we would plot the data. And what we would get, and we need to key it out so people can see it, our red line is net, and we can see that it mirrors the gross, but it is lower because we're not counting what the organisms, the photosynthesizers, used by themselves. Okay, and we can see that light does have an effect. The more light, the more productive. Okay, might as well finish this out. Can ecosystems be too productive? Can you have too much photosynthesis? Can you have too many autotrophs? Aren't autotrophs a good thing? It just makes more food for the critters further up the chain? Well, this is in Hampton Roads, not very far away from here. Take 64, go east, and you'll be there. This thing that looks like this incredible, cloudy, crazy thing in the water is an algae bloom. And there's way more algae than can be consumed by the critters there, and so that algae will decay. It will die. You can see that there's layers, and the stuff up top is going to block the light from the layers down below, and they're going to die. They're going to be degraded by bacteria, decomposed, and the decomposers will use up oxygen. And if you'll recall, we call that eutrophication. Eutrophication. There's a nice word for your review. So this is what I just talked about. Okay, dead zones are caused by eutrophication, and that's a big concern in the Chesapeake Bay. When you have a dead zone, the bacteria decompose the dead algae and basically suck all the oxygen out of the water, so the fish suffocate. Not a happy thing. You might want to remember why algae might bloom in this way. Normally, limiting factors are in place to keep control over growth, but if we supply something that should be limiting, like nitrogen or phosphorus, that shouldn't be plentiful in the environment, and we supply it through fertilizer runoff from the soil, then we encourage algae growth, and then we have a big problem. And that's what causes a lot of these algae blooms and dead zones. All right, that's much longer than I hoped it would be, but there you go. So thanks for watching, and good luck on your exam.